My name is Deandra and my partner is Amrita. We would like to present to you our findings regarding waste management by investigating the extraction of amylase enzyme from fruit wastes to enhance the production of bioethanol from carbohydrate waste.
Our country, Indonesia, is rich in resources, yet we are still keeping up and maintaining the accumulation of organic waste, which marks up to 190,000 tons. This becomes a concern as the rotting of organic waste will heighten the concentration of methane gas in the air, which induces global warming.
One of the solutions that we found interesting was the synthesis of bioethanol starting from organic waste. Bioethanol is defined as a renewable fuel source which is biodegradable and clean.
We decided to conjure an experiment by turning this issue into an inspiration. We developed a strategy in a localized context by using peels and fibers of traditional fruits in Indonesia. We found great potential where the fruits contain amylotic properties that could break down carbohydrate into glucose. With proper extraction, the amylase enzyme can speed up the production of bioethanol to a significant extent. Thus, we began with a research question. How do different types of Indonesian fruit waste like jackfruit, rambutan, and mangosteen affect the activity of its enzyme extracted and its ability to catalyze the process of bioethanol production from other food waste? The amount of enzyme is directly proportional to the amount of protein present in the composition of a fruit. This is because enzymes are essentially proteins. Jackfruit has a protein percentage of 2.4%, mangosteen has a protein percentage of 0.71%, and rambutan has a protein percentage of 1%. A higher concentration indicates that there is higher activity within the enzyme.
We assume that a higher protein percentage found within the fruit will lead to a higher concentration of enzymes produced, which in return will affect the yield, a greater yield, and the purity, speed of the fermentation process, which will produce bioethanol.
Correspondingly, our independent variables involve the different types of fruit waste in terms of their peels and fibers of traditional fruits, which includes jackfruit, mangosteen, and rambutan, while the dependent variables include the activity of the enzyme extracted and the volume and percentage of bioethanol produced. As seen on the presentation, these are some of the controlled variables, but are not limited to this list.
For the prelab, we prepared crude extracts by blending, diluting, and filtering peels or fibers that might contain possible enzymes. We also prepared agar solutions and sterilized equipment for incubation.
Qualitative data was collected by looking at an agar suspension on a petri dish. There were 5 wells, 3 were filled with the enzyme of the fruit extracts, 1 was a controlled negative that was filled with water, and 1 was a controlled positive that was filled with industrial enzyme.
We also conducted a quantitative test using spectrometer and absorbency. We used a vortex mixer to mix Rochelle salt, dinitrosalicylic acid or DNS, and glucose to define the glucose breakdown and create a standard curve using the spectrometer and the cuvette.
This was done 5 trials each for each solution. The fourth phase was the application phase of the experiment. We performed a fermentation process with a diluted rice solution that was taken from the leftover fruit and we mixed it with 18 grams of yeast and 30 ml of our enzyme extract. The fermentation process lasted for 5 days, and after that, the fermented solution was placed in distillation, which was done for 3 hours, and then bioethanol was produced.
Qualitatively, the microtubes, which has a series of colors, show that the darker the color, the more glucose is broken down from carbohydrate. Jackfruit is seen to have the darkest color within the microtubes. In the second picture, clear zones around each well defines the starch agar that has been digested by the enzymes. It is shown that jackfruit has the largest clear zone, which is compared to rambutan, which has the least visible clear zone.
Quantitatively, we collected a standard curve that acted as a tool to convert the results of the absorbance into levels of glucose concentration. Note that the higher the absorbency, it means the higher the glucose concentration. And as seen here, the second table shows the absorbance result of the rambutan solution.
And fourth table showed the absorbency of mangosteen and jackfruit. Jackfruit shows the highest absorbency up to 5.490, which then parallels to the qualitative result how it is the darkest in color within the microtubes. This indicates that a lot of glucose was broken down by the jackfruit enzyme from carbohydrates,
which indicates high enzyme activity within the fruit. 3 trials were conducted to assess the yield of ethanol, which again supports the preceding data, where jackfruit collected the highest average. This further shows the effectiveness of the enzyme that acts as a catalyst at a given time span. The first graph shows the standard equation to be applied, and this graph shows a direct relationship between absorbency and levels of glucose concentration. The second graph shows the concentration of glucose built by the standard curve. This shows that jackfruit has the highest glucose concentration, which proposes highest enzyme activity, while rambutan showed the lowest value.
This was an example of the ethanol percentage from jackfruit solution and fermentation using an ethanol sensor.
The fourth graph shows the average alcohol percentage from the fermentation and distillation process using 3 different fruit extracts. This graph shows how jackfruit had the highest concentration of bioethanol, while rambutan had the lowest.
Data shows positive correlation as well as high R-squared values, which then supports the hypothesis and answers the research question.
However, we're curious on how to preserve our enzyme, so we thought of the idea on making immobilized enzymes.
Immobilized enzymes can be beneficial as they can be reused. Conditions can be manipulated in order to achieve optimum condition of the enzyme.
Though we did successfully create a physical shape from these enzymes, we were unable to sustain its form. This was due to the lack of equipment provided in our lab. It required the use of a freeze dryer, which we didn't have. In the final conclusion, this experiment has taught us to be creative. This solution can be applied to countries worldwide. Normal fermentation process takes up to weeks, but this experiment proves that it can be done in the span of only 5 days. Thus, this experiment shows the plausibility of rapid production of bioethanol at a given time span. This can reduce more waste and produce more renewable energy efficiently. Thank you for listening.